Hello, good morning. Okay, let's do this question first. This is from the last lesson. So we have learned this counter current multiplier mechanism. Just to summarize, what is again a counter current multiplier mechanism? This is loop of Henle. And in a loop of Henle, this is the direction of the fluid at the descending limb, and this is the ascending limb. So it's going this direction and this direction, and the blood is flowing from the opposite direction, right? So if you take these two, they have opposite direction of flow, and if you take these two, blood and the fluid have opposite direction of flowing, and if you take these two, again it's opposite direction of flowing. So the word counter current is derived from this opposite direction of flow. One is going this direction, another one is going this direction, so it's called counter current. Current is the flow, counter mean the other part, right? So it's again opposite direction. And multiplier mean because of this counter current direction, it is increasing the gradients. It can create a gradient, so it can increase gradient is the multiplier mechanism. And we learn the important thing: descending limb only permeable to water, then the water is going out. The water is going out from the filtrate. I think we haven't completed this one, not sure. Okay, so water is going out from the filtrate. When the water is going out, the filtrate is concentrated. It's getting concentrated because when water is decreasing, more solute stay in the filtrate. So then it's getting very concentrated. And this one, ascending, it is only permeable to sodium and chloride ion. You know the sodium and chloride ion, not like water, cannot move by the passive process. It is active process, right? So it is an active process, that means active transport. So that need, I think I will, Maybe I haven't done this. Okay, I will do it again. This is the loop of Henle. This is the collecting duct. The fluid is moving this direction because it's a descending arm, and that part is only permeable to water. Water is going out. That's osmosis. And this side, it's only permeable to sodium and chloride ion. Sodium and chloride ion moves by active transport. Okay, then what will happen? The fluid which enter at this point has high amount of water, and the water is reabsorbed along this descending limb of loop of Henle. When the water is reabsorbed, volume reduced. When the volume reduced, the solute concentration increase. So we take example. So 10 liter of water, the fluid enter at this point. And let's assume some arbitrary unit of 1000 solute. Now when it comes to this point, so 8 liters of water is reabsorbed. So what is the amount of water? 2 liters. But the solute, same. Right? The water is 2 liters and the solute is 1000. So if you want to calculate the concentration at this point, so what we can do, 1000 divided by 10, it is 100. It's 100 solute per liter. Here we calculate 1000 divided by 2. So it is 500 per liter. Now you can understand the concentration is 100, and here 500. So it is concentrated now. So when it moving upward now, the solute is further moving out. The solute is moving out, right? So when solute is moving out, maybe the solute, when it comes to here, it was 1000 here, and it was 1000 here. Now when it comes at this point, it may be 200 arbitrary units. Solute is now 200. The volume is 2 liter. Then again it become 100 arbitrary unit per liter, right? So it's again become 100 now. So what happened? Excess solute accumulate in this medullary or interstitial space. Now again, this continue through collecting duct. This fluid is going through the collecting duct. When it comes to this point, still some amount of water can go out from collecting duct because this side solute concentration is high. And you know, the theory of water potential, high water potential to low water potential. So this area has low water potential, then the water is going out. So at the end, maybe it is a 1 liter uh, volume passing out as urine, 1 liter. So direction of blood flow is also like this direction. So water can again enter into the blood. Okay. Now, if we have to answer a question, ask relevant to this kind of information, what are the main things we should highlight? Now, we should highlight this point. Even it doesn't include in the marking scheme minor details, we should include this point, right? Descending limb is permeable to water. As a result, water is leaving out. Ascending limb is only permeable to sodium chloride. Then the sodium chloride move into the medullary or interstitial space. So this is the medullary space, right? So we can write 1 point. I don't count as a point to get a 1 mark, but just write water is moving out from descending limb by osmosis. Water leaving out, I think it's important, right? So uh, from descending limb by osmosis. Sodium chlorides pump out by active transport along ascending limb of loop of Henle. This establish a gradient which is known as countercurrent multiplier mechanism. Right? Countercurrent multiplier mechanism. Now, what I did so far, I actually introduced what is countercurrent mechanism. Right? So first, you see the red dot here, the first one, what is moving out by osmosis, number one. The second, sodium chloride is moving out by active transport, the ascending limb into the medullary space. So this create a gradient, this help to create a gradient which is known as countercurrent multiplier mechanism. Then the second important thing is the medullary space, sodium chloride concentration is high. As a result, when the fluid enter into the collecting duct, still some water can go out by osmosis from the collecting duct into the medullary space. So this could be the third important things to mention, right? One, two, three. There's another one, fourth one, but we can't learn it here. We can add still the fourth information after we learning it, right? So one and two consider as a countercurrent multiplier mechanism, and the third one is again important, that water can go out from the collecting duct. So we need still to add the fourth one, which we're going to learn in the next coming section, right? Okay. The kidney controls blood volume and plasma concentration by regulating the loss of water in urine. Some mammals have adapted to their kidneys that enable them to live in a different habitat. Brown rat, urine concentration is 2900. Mitochondria in the thick ascending limb 3.6. Sodium ion concentration in the extracellular fluid in the medulla 45. Kangaroo rat urine concentration this much. Mitochondria this 95. Okay. Uh, One important point we should remember, kangaroo rat has a long loop of Henle, right? So kangaroo rat, a long loop of Henle. I think this is something to remember, because this question relate to kangaroo rat asking many times, and we should know that kangaroo rat has a long loop of Henle, because I think the textbook this as a given as a good example, so then we know kangaroo rat has long loop of Henle. Now if you look this question, deduce why the kangaroo rat is more successful than the brown rat living in desert habitat. Okay, even though in this question it is not mentioned to use the data in the table, deduce why the kangaroo rat is more successful, of course we have to use the data in the table, because this question is given to use the data in the table. And another important thing, when information given in the table, you should use all the data. So you can consider as a rule, should use all the data in the table. Sometimes it may be rarely, it is not important to use one or two data in the table, but almost all questions coming in any unit, we should use all the data in the table. Doesn't matter whether they are in the marking scheme or not, because our chances are high to get the full mark, right? Right now, when we're going to answer this question, Why the kangaroo rat is more successful than brown rat at living in a desert habitat? So we have to first cue the most important introduction, that is desert habitat has less amount of water, or water availability is less in desert habitat. Therefore, mammals living in desert need to conserve water. And kangaroo rat has a long loop of Henle. Now this is the introduction part for this question. Now, if you look at the marking scheme, they offer one mark saying that the water is less in desert habitat, right? So you get a one mark for this one. But there are other years when they ask similar question, they give one mark for this one. Kangaroo has a long loop of Henle. But this particular question, you get only one mark because this is not allocated one mark for this one. But we should remember to add certain additional information because we never know what is exactly in the marking scheme. It may be there, it may be not there. Right. And no harm. We can quickly write these two sentences. And then we started to use in the data, in the table, what we're going to say. Uh, okay, anatomy. Then we can write kangaroo rat produces highly concentrated urine because urine concentration is 5,500 and this one is 2,900. I mean, if you want, you can write, just make it perfect, which is 5,500 milliosmol for DMQ. Then we can write this, the ascending loop of Henle. So I told you the ascending loop of Henle can pump sodium and chloride ion by active transport. Active transport need energy, ATP, produced by mitochondria. Now it has high amount of mitochondria. So we can write that point, kangaroo rat pump more sodium chloride from the from the ascending limb as it has more mitochondria. You can write so does ATP. More ATP, right? More ATP. So now we also address the brown rat and kangaroo rat is better because it has more ATP. And the last point, the medullary region, sodium chloride concentration is also high. I mean, the medullary region is this place, right? So then you can write the last point because medullary region has high uh, ion, sodium ion, right? They use sodium ion only. Sodium ion, water still moves from collecting duct to the medulla. It's 95, it's higher concentration, so it can still move from the medulla. So here, even though 3 marks, we wrote a little bit more, but we address these 3 points, right? This, and our introduction. So this, we try to get 3 marks if we aim like this kind of writing skills. Okay, I will send this document and you can get it when you want to write. Right, this is the another lesson. This lesson is important for the fourth point for this. So I mentioned this countercurrent multiplier mechanism. Number one, water moves by osmosis descending limb. Sodium and chloride ion moves by active transport ascending limb. And then the medulla has high sodium chloride. At the fourth point, water still go out from collecting duct, and there we have a small mechanism to explain at the fourth point, right? So we're going to learn it in different topic because it is related to many questions. Osmoregulation role of ADH. Right, ADH is anti-diuretic hormones. ADH. Next, when it comes to osmoregulation, I think some of the lesson you have learned previously is level hypertonic, hypotonic, isotonic like things, and osmosis, water potential. They all important to understand the concept of osmoregulation. All right. If we take human body or other animal body, the cells, all the cells communicate with extracellular component. There's something called intracellular and extracellular. So this is inside the cell, intra. This is extra, extracellular and intracellular. What is intracellular? Is the cytoplasm. Cytoplasm is the intracellular area. Extracellular. Now, extracellular include several components, right? So, what are the components? The fluid between blood and the cells. Let's assume this is the blood and the plasma. So we can call this as a tissue fluid. Now there should be a correct balance between extracellular fluid and intracellular fluid, because the basic concept what we have learned AS level, if the water potential high in outside, it's really high, the water moves out, then the cells are shrinking. Maybe other way, right? Sorry. If the water potential is high outside, the water is moving in, because this side water potential is low. Then the cell can burst and it can rupture, right? On the other hand, if the water potential is low and this side water potential is high, what is going out? This lead shrink of the cell. This concept we learn AS level, isotonic, hypertonic, and hypotonic solutions like that. So it is better to have isotonic conditions. So isotonic condition is the correct condition. Then there is a no net movement of water, but separate mechanism can be operated from the blood to send fluid out of the blood, and then the cells can take it. Now, day-to-day basis, this concept is not easy to maintain, because if a large volume of water taken out with less solute, then the water potential is increasing. So let's say if we drink quickly 2 liters of water, then the water potential is really high. If we drink 3 liters of water within like 30 minutes time, the water potential is really high. And if we drink water with solute, then solute and water potential is in the right concentration, then there is again a little bit of balance. But most of the time what will happen, either we lost water because of sweating in a very hot day, or maybe vomiting, or maybe diarrhea like condition, we can lose water. If water lost, the body try to regulate by taking absorbing more water from the kidney, right? So we can understand now, if we lost water by sweating, the body has mechanism to make concentrated urine. If we take large volume of water, kidney try to excrete water with urine. So kidney play an important role maintain this water balance. This is osmoregulation. Keeping constant amount of water inside. That's a homeostasis. The basic concept is keep internal environment constant. Keep water level constant in the body. It's the osmoregulation. Now we're going to learn how this mechanism take place in the kidney. Okay, we start from the osmoregulatory center of the body. Where is the osmoregulatory center? Again, this place is we can find it in the hypothalamus pituitary area, right? So this is hypothalamus and this is the pituitary gland. Pituitary gland has two parts. One is the front side, other one is the back side. So we call anterior and posterior. The back side is referred as posterior pituitary and the front side is anterior pituitary. Okay, what is pituitary gland? Pituitary gland is collection of cells which are more specialized to produce hormones. So these cells are specialized cells to produce hormone. What is hypothalamus? Hypothalamus is it's towards the brain. That means they have nerve cells, neurons, right? So hypothalamus is basically consist of neurons. So these represent neurons. And these neurons are specialized to detect external stimuli, like receptors, right? So there is a area in the hypothalamus which has collection of neurons. So I just draw like this, and these neurons consider as osmoregulatory center. So specialized neurons make this osmoregulatory center. Hypothalamus always communicate with pituitary gland, and sometimes these neuron cells ended up in posterior pituitary gland. Now, hormone ADH, a little bit special in this case, ADH is produced in hypothalamus. Otherwise, most of the hormones produced in the pituitary gland. So this is a little bit of exception that ADH is produced in hypothalamus and this is stored in the posterior pituitary gland.